Okay, well, thanks very much for that introduction, and thank you for the opportunity to come and speak at this very important occasion, the opening and the launch of the centre. I work kind of more at the front line. I work with policymakers, people in the Ministry for Primary Industries, Ministry of Health, for example, and really try to use some of the tools that Alexi's already mentioned and others in the room have developed, such as David Bryant as well, in real-world settings to try and solve problems. And I'm going to talk about one example today. I'm going to talk about the control of a particular foodborne disease. Happens to be the most important one in New Zealand. It's the most prevalent one, and it's one for which New Zealand has a particularly bad reputation. And the disease itself is called Campylobacteriosis. It's caused by a bacterial species, Campylobacter, and it's still at the top of the list of notifiable diseases in New Zealand. And it's important as a cause of what we call enteric disease, so this is vomiting, diarrhea, and can lead to hospitalisation in people. We have around about 6,000 cases a year. Other foodborne pathogens we're also working on using similar techniques are also in this list of the most important notifiable diseases in New Zealand and around the world. Between 1980 and 2006, we had a growing slow burn epidemic of Campylobacteriosis in New Zealand, and our reputation internationally was getting worse and worse and worse. This is New Zealand tracking against other similar countries around the world. In 2006, there was a landmark paper in the New Zealand Medical Journal by Michael Baker, a prominent public health researcher and physician, saying, we know it's chicken that's responsible for this. We should remove from the sale of all fresh poultry, freeze it, and that will help to reduce the epidemic. There was a growing body of evidence implicating chicken. But at the same time, there were a lot of other people that were thinking there were other sources of infection. You also find this bacteria in cows and sheep, you find it in wildlife, so there were other potential sources of infection. So there was a lot of confusion around about that time as to where people were getting infected from. And knowing that was such an important public health question, it really needed some of the best tools and technology applied. So in 2005, we set up a Sentinel surveillance site. This is the only Sentinel site in New Zealand for any disease, I think, but particularly for Campylobacter infection. And what we did was we had all the human cases that came into the Manawatu region, came through our laboratory. We cultured them, we typed them using the same methods that we typed isolates that we got from chicken, bought from supermarkets, from red meat, from environmental water, and from the catchment areas of the environments that they were sampled from. So these are high-use recreational swimming spots that we sampled from at the same time. And this has now been running, well, actually it's still running now, so for 11 years, we're in the 12th year of the Sentinel surveillance program. And this is the kind of population structure that you see. This is actually using a tool developed by one of David's postdocs, Jessica Lee. These are all of our isolates typed using multi-locus sequence typing. Multi-locus sequence typing is just a 7-gene sequence typing scheme. It's a very small part of the genome, but it happens to be useful because you find a very strong correlation between certain strain types and certain hosts. We can also see some that are found in wildlife and in water. This is one that's found in the pukeko and water rails. Never turns up in people, but is still the same species. This one here was very important. This is sequence type 474. That was the New Zealand epidemic strain for the early part of that epidemic going up to 2006. It accounted for about a quarter to a third of all human cases, and at the time we didn't know what the source of that particular strain or any of the other strains was. So we developed a framework which has been published now in WHO documents and also in the peer-reviewed literature, where we're essentially trying to find out through a number of different investigative tools what is the source of human infection, and that informs the policy decisions. So we've got our animal amplifying hosts, or reservoirs, and for this we use the technique of reservoir attribution, which I'll talk about in a minute. Then we might use other modelling techniques to identify what pathways these animals infect people through. What are the precise exposures and what are the precise risk factors? And you might use epidemiological studies such as case control studies to infer those. So this is the broad framework we use, and I'm going to be talking about this particular approach of what we call reservoir attribution. Now, since then, the tools that we were using then were relatively crude. Whole genome sequences weren't as readily available as they are now. And so we've kind of revised this, and this is a recent paper with Tim Vaughan from Alexi's group, where we're actually incorporating some of the newer whole genome sequencing technology and some of the evolutionary models that Alexi was talking about, such as the phylogeographic model, and the structured coalescent model, all of which are now being used to infer where humans are getting infected from and what those transmission pathways are. It was developed for Salmonella. The model that we used in our Manawa 2 Sentinel site, we used a number of models, but the one that was based on the coalescent model was also one based on the Asymmetric Island model. And essentially what you're doing is reconstructing the genealogy based on the sequences of Campylobacter that you've got from your different sources. You estimate recombination, mutation rates, but really what you're interested in is the relative migration from these sources into the human island, and that gives you the relative contribution of those sources to the burden of human disease. And this was a model that was published originally by Danny Wilson, who we subsequently collaborated with. And this was the answer that we got fairly early on. It was quite clear that no matter what modelling approach you used, the chicken was indeed responsible for most of the human infection. In fact, one particular poultry supplier, supplier A, was certainly responsible for most of the infection that we were having in the Manawa 2, and as it turned out, across New Zealand as well. Now that, in a very quick summary, was where we were up to at that point. That resulted in implementation of policy. This was presented to the policymakers, to industry, the poultry industry. The industry then complied, opened their doors to each other, and the risk management strategy was put in place. Now, the effect of that was to reduce the level of contamination on the chickens you buy in the supermarket, and that had a very dramatic effect on public health. So within the space of a year, we went from 16,000 cases to around about 8,000 cases, and the hospitalization rates also declined quite dramatically over that time period. And it's estimated that alone, through fewer people getting sick, fewer people hospitalized, fewer people taking time off work, fewer people with children that were sick having to take time off work, is estimated to save the New Zealand economy around about $50 million per year. So if you can get the decisions right based on good science, it can make a real difference to not only whether people get sick or not and the misery that people suffer, but also it can save the economy millions of dollars. So as I say, this was a 5-year target that was originally set, but it was actually achieved within a very short period of time. You can see the seasonality, and it's trucking along now. We have around about 6,000 to 7,000 cases per year. Now, sequence type 474, which was our epidemic strain, not surprisingly, also declined very rapidly over that time. We now model urban and rural areas separately using the same models that we developed before. But this one we were looking at was obviously clearly rapidly evolving. Although it was found mainly in one poultry supply, later on in the epidemic it was found in cows, in sheep, in wildlife, so it seemed to be evolving through time. The question was, what's going to happen next, and were we waiting for another epidemic strain to come into New Zealand, as we often see with these types of pathogens, which was then going to take over from 474 and cause more problems. And we did. In 2014, we saw the arrival of a new strain, which was called sequence type 6964. And the reason why this was so important was because it was not only a Campylobacter strain that was causing problems in humans, it was also chicken associated, but it was also resistant to 2 antibiotics, tetracyclines and fluoroquinolones. And the reason why that was important wasn't necessarily because those antibiotics were being used to treat the condition. Ideally they shouldn't be. But it transformed the New Zealand landscape. We went from being a country where we had relatively low levels of antimicrobial resistance to one with moderate to even high levels of antimicrobial resistance over a very short period of time. And this was the kind of news reporting that we saw at the time. Although technically they're not superbugs, they were being labelled as superbugs, and it was a very emotive issue. Television, newspaper reporting was very prominent at the time. So the emergence of 6964, we saw the first 2 human cases in our Sentinel site. These are the first ones to be reported in New Zealand in August 2013. One of the unusual things was that it was also found in all three of the major poultry supplies in the North Island, not in South Island, but just in North Island. So it had already infected the poultry supply, but not just one company, but multiple companies. And it was also in the parent flocks. These are the flocks that breed the chicks, that then, over a period of just a month or so, become the broiler birds that you buy in the supermarket. As I said, these were resistant to tetracycline and fluoroquinolones, and we then ended up with going from a 0 to almost a 40%, 35 to 40% of positive birds. So we saw a sharp increase also in human cases across New Zealand as well. A similar study was done that showed that this was now the dominant strain in people. And if you ever feel like eating chicken liver, I wouldn't advise it, because it was also responsible for a major outbreak, yet one of a number of outbreaks associated with chicken liver pate. It's one of the most dangerous products known to man. So ST6964, interesting because it's a member of a particular clonal complex, which is a chicken-associated clonal complex called 354. So it's one of a cluster of sequences. And the only other isolate in the international database was from China. Now we know we're actually trying to source the isolates from China to see if we can compare them with the New Zealand strain. How it got into New Zealand is an interesting question that we've yet to answer. But when we look at using whole genome multi-locus sequence typing, so this is looking at basically the whole core genome and comparing it with other isolates that we have in New Zealand. This was our previous epidemic strain. This is our new resistant epidemic strain. We can see it's quite different to anything else that we'd seen before. So the key questions that we wanted to address using the kind of approaches that Alexi was talking about was, what's the genetic basis for resistance? How long has it been in New Zealand? How has it been transmitted between those different poultry supplies, given that they're supposed to be but strict biosecurity and very limited movement between them? What mechanisms have driven the emergence of this particular strain, and how is it evolving? There are a whole range of different ways in which bacteria evolve, and I'll come on to talk about those in a second. In fact, now. So Campylobacter is a bacteria. It's a spiral-shaped bacteria. It's got two flagella or propellers on each end, and it's believed that it uses that to spiral its way through the gut mucus, so it kind of repels its way through. And the cell surface in particular and the flagella are highly immunogenic, so these are the molecules that are often recognized by the human immune system. And so it's evolved a number of different ways of trying to avoid and adapt to particularly changing environments, changing environments in the guts of animals and in people as well. And the tools that it uses simple point mutations to create non-synonymous changes which can change proteins and protein functions. It can use recombination, so this is horizontal gene transfer, where whole pieces of DNA are moved from one organism to another through a range of different mechanisms, including bacteriophage, and the use of conjugation or plasmids, which move into the cell through that route. We have other methods, slip-strand mispairing. This is where you have sequences, long sequences of the same type, poly-Gs, poly-Cs. They slip and cause these switching events that can turn on and off genes very effectively. And we also have other things like methylation of both DNA and of proteins. There's a whole range of different tools that they use to avoid the immune system and to avoid phage infection and all other kinds of things. And this creates this vast array of potential phenotypes that are available for natural selection. So we've done some previous work with Paul Fernhead at Lancaster University in particular, looking at two of those mechanisms, mutation and recombination. And what we found was for Campylobacter jejuni, the species that we're working with, actually recombination per event was much more likely to happen than mutation. And because it brings in so many more differences, it was more important for the evolution. But the trouble with horizontal gene transfer, or recombination, is that it confuses the phylogenetic signal. You've no longer got a colonial relationship between isolates. Each gene, or each bit of a gene, or maybe even each nucleotide, may have a different evolutionary ancestry. And so we need to think of new tools that can help us make sense of that information. And although I'm not going to talk about this in any great detail, this is some work I've been doing with Tim Vaughan and Alexi and David, looking at developing another package for use in Beast 2, which enables you to jointly model the clonal relationships and also what's called the ancestral recombination graph, so jointly modeling the horizontal gene transfer events as well. So how have we approached this new strain? Now, this new strain, the resistant strain 6964, we are now able to use whole genome sequencing. And one of the things that we did was genome sequence an initial group. This was around about 90 isolates that we had from different poultry supplies and from humans. And if you just extract the SNPs using this particular tool, Snippy, and draw a neighbour net tree, again using the split tree software developed by David and colleagues, you can see this kind of pattern, and you see a very strong association between poultry supply and clades A and B. The red are one particular poultry supplier A, and down here, other than this red one here, you've got suppliers B and C, all interspersed with the human infection ones as well. And interestingly, that split there is associated with one particular part of the genome, one integrated element, and this one due to another one. And what we find was a high density of these particular changes that are occurring in what are called prophages, which are bits of genomes that are inserted into the bacterial genome from bacteriophage. So they're called integrated elements, so they come from viruses that infect the bacteria. So we saw quite a lot of information that was suggesting they were important in the evolution and separation into the different poultry supplies from this one to this one here. Using the particular sequencing tools that we were using, Illumina sequencing technology, we had a problem. And this was identified when you looked at the read depth of the reads from the sequencer. You found that there were certain areas associated with these phage elements that were double the density in other areas as well. And when you looked at the raw reads, you could find evidence of two different alleles, each around about 50% frequency, suggesting that they were mapping on top of each other. So we needed to take a different approach if we were really to understand what was going on here. And that's where we use newer technology, which is the single molecule technology, or PacBio sequencing. And what we did was we selected an isolate from each of these clades, and we can now use what's called reference guided assembly by combining the Illumina reads with the whole single molecule technology to get much better inference about what's going on in these particular genomes. And this was done in collaboration with the University of Melbourne, the Melbourne Diagnostics Unit, and Debbie Williamson, who's based there. And one of the things that it revealed, which was very interesting, was that you had, compared to a reference strain here, these integrated elements. So these are ones that come from phage, and this one here is present in all four and inverted relative to the reference strain. But we also had another one which showed very strong homology to this one, but seemed to be inserted at 3 separate sites within 3 of the genomes and absent from the other ones. So you see a lot of variation, which is quite interesting. And these variants, in particular that variant that was inserted at the 3 separate sites, is highly variable. So this suggests that even though we think it's only happened over a recent time period, all this evolution, we've actually seen possibly 3 separate events introduced by bacteriophage of similar sequence, but variable enough to be different. So that's transduction recombination we see. We also see evidence of just straightforward recombination occurring in areas which are involved in immune avoidance. So this again is a particular area that we're interested in. We also see a plasmid which has come in, and this plasmid is what brought in the tetracycline resistance, the antimicrobial resistance. But the remnant plasmid, which has been, the genes have been excised and put into the chromosome, is still present in these 2 clades, but absent from these 2 clades. So we're seeing a difference that is occurring there. Stripping away all of the recombination, we can then use some of these tools, although it's only based on a small data set at the moment, to try and estimate the time of common ancestor. And although there's very wide confidence intervals, it's consistent with a relatively recent introduction. And again, wide confidence intervals, but the suggestion that there may have been a population increase around about early January, around about that particular time. And this is where the plasmid's been lost here, and these are all found in one particular poultry supply. We also see evidence of this slip strand mispairing. So these are the on-off switches that are occurring through these poly-G tracks that we see quite frequently. So the point I'd like to make here is that we're seeing just about every possible mechanism in action in this particular bug. And this is an important bug from a public health point of view, so we really need to get an understanding of what's actually driving this. So key questions. What's the genetic basis for resistance? We can use the sequence to identify that it's actually that plasmid that's inserted the tetracycline genes. We find it's a single mutation in one gene that's conferred the fluoroquinolone resistance. We estimate that it's not been in New Zealand that long, so just a few years, although that analysis is complicated by what's going on in the single sequence repeats of mobile elements. Been transmitted, we can see strong evidence for potential shared parent and grandparent stock that have resulted in that transmission down through the different lines. What's driven the emergence? Fluoroquinolone isn't used in the poultry industry, but tetracycline has been used in the parent stock. So we think that may have resulted in large-scale expansion of this particular clone at the expense of others. But what's driving the evolution is really complex. So it's a situation where you have point mutations, you have different types of transformation, horizontal gene transfer, phage-mediated transduction, conjugation, single sequence repeats. They all seem to be acting in tandem to create this huge phenotypic variation which is occurring in this particular strain. And we could only get this information through the use of whole genome sequencing. This would have been the same strain if we'd used classical typing methods. So just to conclude, so using these kinds of tools and techniques, we have got a better understanding of the epidemiology of a lot of these pathogens which are important causes of disease in New Zealand. We use models which are based on evolution, such as a coalescent model, to make inference about these. We're applying it to a whole range of different pathogens, but we may need to rethink, particularly with these very complex and interacting evolutionary mechanisms, about how we really understand what's driving evolution. The tools have advanced. We've had this situation where we identified poultry as the main supply. That did result in a major decline, but we have had this emergence of this new antimicrobial resistant strain that we need to tackle. We need to use this complete toolbox approach, so combining epidemiology with spatial modeling, with population dynamics information, with population genetics and other types of models. And we're expanding our isolate set for the current data set using more sequencing. We're going to start a big case control study in a few weeks' time, working with industry and with government as well to try and reduce the prevalence. And the need there is always there for better models and better tools for us to make sensible inference, to make good decisions, to inform public health decision making. Huge number of people involved in all of these projects going back over time and funded by different sources as well. Thank you.